Today I'm talking about extracellular reference profiles, which has been a project we've been working on in the first phase of the ERCC and have been continued until now. Our previous work was focused on a limited set of normal healthy individuals who donated cell-free plasma and serum, 6 males and 7 females, collected at 12 time points over 2 months. Since this is a hands-on workshop, what also like to focus on the methods. We've used our sRNA-seq analysis platform mainly for this project as well as for the continuation. The analysis were mainly done using small RNA atlases and R. In R, we used unsupervised clustering, differential expression analysis, and seq. In our previous results, we observed noticeable differences between serum and plasma, as well as minor differences between genders. Also, we discovered a novel extracellular RNA phenotype in one participant who had a twofold elevated microRNA concentration and overall a signature of likely neuroendocrine origin defined by microRNA 375. This phenotype persisted over 2 years. In our continuation, we now included more participants, roughly 230 normal healthy individuals, 63 males and 173 females. We collected samples at 2 time points over a time of on average 2 weeks. In order to have a distinctive phenotype, we also included plasma samples of 21 pregnant women at different stages of pregnancy. And in a third cohort, we included the individual that has the neuroendocrine phenotype and his family. We added more analysis methods, correlation analysis. The lab also uses occasionally dimensional reduction like PCA analysis and TSNE. However, this is not the main focus of the work. Now, if we do unsupervised clustering of the representative sample set as shown here, which is constituted of about 250 individuals, we see that the main sample groups separate well using this method. On one extreme, we see the pregnancy samples, which are distinctive from the majority of the normal healthy individuals, although we actually identified one individual who turned out to be pregnant within this group, which is, I think, a good validation of the concept that was unexpected, but pregnancy tests confirmed her pregnancy. On the other hand, we see a distinctive branch of all the samples of individual P12 collected in the first 2 years, which are probably the most extreme phenotype. Now, in order to define normal variability amongst the group of the normal healthy individuals, we did classic statistical analysis and computed the quartile coefficient of dispersion, which essentially relates the interquartile range to the mid-hinge, which is something like the median normalized count. And there's a nice there's a nice function that can be fitted, suggesting that the overall quartile coefficient of dispersion is more or less a function of the overall abundance, which is probably due to technical limitations of the method. Since this can also be seen as something like a standard deviation, it also suggests that at high read depth, at very abundant microRNAs shown here, we have about a 20% variation up or down. This approach also allowed us to determine thresholds for what is normal variability and what is elevated variability. And we did this for the 344 female data sets, including the one pregnant. We did this also for the 126 male normal data sets. And we did observe a group of microRNAs, which is partially overlapping, which shows higher than usual variability in these two groups. Now, we asked the question whether these highly variable microRNAs actually are correlated, and did a classical correlation analysis using Spearman correlation coefficients, which identified that some of these highly variable microRNAs, here shown in bold, all the bold ones are the highly variable ones, are in fact correlated. And these involve liver microRNA 122 and 885, as well as muscle-specific microRNAs. These are likely released from the heart. Dr. Kemal Akkad has shown that these so-called myomers are highly elevated in patients with heart failure. In addition to that, we observed that some of the highly variable microRNAs, including neuroendocrine microRNA 375, are correlated with certain epithelial microRNAs. These are actually all elevated in individual P12. They also seem to form one correlated group. And we observed two novel groups that we hadn't seen before, a neuroendocrine 2 consisting of cistron 134 and cistron 127, which is likely originating from the pituitary gland, and another correlated group of microRNA 33B, likely released from the adrenal gland, and 205, another epithelial contribution. Now, after having seen this, we looked at how actually the different metadata groups compare by differential expression analysis. And this is shown here as an example for the gender-related microRNAs. As you can see, the differences between men and women when it comes to the abundance of microRNAs in plasma is really small. And if we use the fitted QCD limits, it's actually in the range of what is to be considered normal variation. So we don't see there is a lot of biological significance here. There are two exceptions. One is microRNA 133B, which is a myomer, which appear to be about twofold elevated in men, and a neuronal microRNA 124, which appears to be elevated in women, although this is both close to the limit of detection. More interesting are the phenotypes we included as discernible, which are the pregnant women. Here, the contributions when comparing, for example, trimester 1 samples are quite distinct from the normal healthy controls. Unfortunately, there are not that many tissue-specific contributions, except maybe neuroendocrine contribution 134, cistron 134, 43, as well as placenta-derived cistron 498. When looking at trimester 2 and trimester 3, the highly significant differences involve mainly cistron 498, which is placenta-derived. And when looking at this box plot, one sees that this contribution actually increases about 100-fold when comparing non-pregnant to trimester 1 and 2 women, and about up to 5,000-fold when comparing trimester 3. So this fits nicely to the biology. Using these microRNAs that we identified, it is actually possible to discern early and late stages of pregnancy, and somewhat to also discern trimester 2 and 3, although not completely, using unsupervised clustering. Now, the question arose whether the individual P12, which had this neuroendocrine signature, was still showing the signature after 3 years. And we had to reach out to him and his family to participate in this follow-up study. And we did, again, small RNA sequencing followed by unsupervised clustering. And the result clearly shows that the signature is still present. And it's only present in this individual and not in his relatives. Now, considering the different sequencing methods we had to apply as covariates, we can clearly say that this individual has this neuroendocrine signature defined by a 10-fold elevated contribution of microRNA 375. And several epithelial contributions involving cistron 200A3, 141, and 205, amongst other differences which are not as strong. Irrespective of the sequencing approach, we had to switch from high-seq to next-seq. The relative abundance of this signature based on 375 is really comparable in the first 2-year samples as well as the year 3 sample. Now we ask the question whether the origin of this signature is actually based on the vesicle contribution of this individual or the supernatant, and fractionated samples we obtained from this family as well as him. And the analysis shows that it's clearly based on the supernatant fractions, which are distinctive from all the rest. When it comes to his when it comes to his pellet contributions, his samples cluster either just next to the his family members or even within the group, but they are not that distinctive as shown here on the right. We also observed that we have a much lower concentration using spike and calibrators of microRNAs in the pellet fractions as compared to the supernatants. And this already considers the fact that we use 10 times the material for the characterization of the pellets compared to the supernatants. Of course, the question arises, what can one utilize when using, or what can one expect when using pellet fractions? Is there an enrichment of certain specific microRNAs? There is, however, at a very modest level. We observed that many of the tissue-specific microRNAs are only about 10 to 20-fold enriched in those fractions. And especially, for example, the neuroendocrine microRNA 375 is highly depleted both in the 10,000 G fraction of microvesicles as well as in the 100,000 G fraction, which are the nanovesicles, including exosomes. When it comes to pregnancy, where we did a similar analysis, we observed that the placenta-specific cluster was only threefold enriched in both pellet fractions, as shown by this box plot here. However, after doing multiple measurement corrections, these differences actually become insignificant. Now, the take-home message is probably in terms of methods, dispersion and QCD analysis methods are very useful to determine normal and elevated variability and define variability thresholds in normal individuals. This allowed us to identify at least 7 distinct sources contributing to extracellular variability. Following up with differential expression analysis, we could stage the stages of pregnancy. We could also confirm that the P12-associated phenotype is still the most distinctive, unique, and constant signature we've observed so far. Using differential ultracentrifugation to enrich for microRNAs in vesicular fractions, we learned that we have a much lower yield in microRNAs and in fact observed the depletion of several tissue-specific fractions and only a mild enrichment of pregnancy-associated microRNA changes. Now, since we've not only done microRNA analysis, but also looked above the horizon looking at different RNA classes, we can say the promiscuous nature of microRNAs actually limits the discovery of cellular origins and phenotypes, and potentially also deconvolutional analysis that aim at the same approach. We would like to characterize extracellular mRNAs, and Ryan fortunately already addressed several of the problems associated with that. These limitations prompted us actually to switch in related projects to RT-qPCR-based approaches, which, however, have very limited discovery potential. Desired would be enrichment and depletion methods for extracellular mRNA sequencing. With this, I would like to come to an end and thank all people involved in this project, in particular people who worked as postdocs and research associates on it, including Kemal Akkad and Edo Bendorf, and Tom Tuschl, as well as the NIH for funding. And thank you very much for your attention.
Thanks, Klaas. If you could give a real quick answer to the question, could you detect fetal RNAs in the serum or plasma of pregnant women?
Using qPCR, yes. Using sequencing, we could only observe increases in hemoglobin transcripts but we did not observe fetal RNAs. And regarding the fetal RNAs, we actually used Y chromosomal transcripts, which should be in any cell type in pregnancies where the women were pregnant with boys. Using qPCR, it was no problem to detect either the Y chromosomal transcripts and also certain placental transcripts.